Hello everybody, Miss Wood here with your notes for Topic 5, Biotechnology. So my expectation is that you have your paper notes with you, that you have turned to page 7 of the notes, and you are following along, getting the notes, and also completing all of the quick checks. So let's go ahead and get started. Again, this is page 7 of your notes. All right, so our next topic on synthetic biology tools and techniques is called recombinant DNA. So when you recombine something, you are combining something, right? And so essentially what we're doing here is we are taking DNA from one organism and we are combining it with DNA from another organism to essentially make a new piece of DNA that has genes from both. So how does this work? Well, the first step is that you have to cut the DNA, and you use something called a restriction enzyme. We learned about enzymes in our biochemistry unit. Remember, enzymes do chemical reactions. The chemical reaction that they're doing here is cutting DNA. So that's why I used this graphic right here. You can see it's got like a little scissors and it's cutting DNA, and it cuts it at a very specific sequence. It just doesn't cut it anywhere. It cuts it at a very specific sequence of A, T, C's, and G's, whatever the pattern is that it's looking for. The reason for this, why it doesn't just cut it randomly anywhere, is that it needs to fit together because we're recombining DNA, right, like a puzzle. And so the puzzle pieces need to fit together when we combine the DNA. All right, so essentially we are cutting a piece of DNA called a bacterial plasmid. So bacterial plasmids are circular pieces of DNA that are in the bacteria cell. And so in this graphic down here in the bottom right, you can see we've got the plasmid and we've cut it. So we've cut that circle. And then we've also cut out a gene from another organism. Maybe this is a gene to make whatever organism our end game is, to make it bigger or to make it more colorful or to make it some trait that's more desirable that we want. And so we are adding that gene to the bacterial plasmid. We then need the enzyme DNA ligase to glue it all together. And so then once those puzzle pieces have been fit together, they've been glued together, we get recombinant DNA, where we have the bacterial plasmid, so the bacteria's DNA, combined with whatever new trait that we want. Essentially, then we can put that DNA back into, say, the bacteria, and then the bacteria will do what we want it to do. So, for example, this is how we produce insulin now. Essentially, the bacterial plasmid has the gene for making insulin inserted, and then we put it back in the bacteria, and the bacteria just make tons and tons of insulin that then patients, diabetes patients can actually use. All right, so the next synthetic biology tool and technique that we're going to be talking about is gel electrophoresis. I know it's a long word, electrophoresis. And what this technique does is it separates the DNA. Based on the size, all right? So we talked about how we can cut DNA, right? We use restriction enzymes. So we can get a piece of DNA and then we can cut it at very specific locations where we find that pattern, whatever A, T, C, and G pattern the specific restriction enzyme is looking for. And then we take the cut DNA and we run it through a gel electrophoresis machine. The machine kind of sorts the DNA. So the smaller segments of DNA move really fast through the gel in the machine, whereas the larger segments take a lot longer to make their way through the gel. All right, so let's take a look at the machine right here. And so how this works is we need a DNA sample, and then we need to cut it up. Remember, it's cutting it at specific locations where a pattern is found. So the restriction enzymes are, added to the DNA, and it gets all cut up into different size pieces. Then the samples are loaded into the top of the machine. These slots are called wells, and this is where individual samples will be loaded into the machine. I also want you to notice that the machine has some charges on it. All right, so it's loaded into the negative end of the machine, and when we turn on the machine, a charge is applied to it, and so there's a negative end here and a positive end down here. So DNA is negatively charged. And I'm sure you've heard that term, opposites attract, right? And so when we turn on the machine, the DNA is going to be attracted to the positive end of the machine, and that's why the DNA moves. So this electric current, when we turn on the machine, it makes that DNA attracted to the positive end, and it will move through. This is a gel. The gel has holes in it. It's basically like an obstacle course for the DNA. Kind of like if you've ever seen like American Ninja Warrior, it's the American Ninja Warrior course for DNA. And so the DNA has to try to work its way through the obstacle course to get to the positive end. The short pieces of DNA, they're able to work through the obstacle course pretty quickly because they can go through the hoops, go through the holes, around the corners. But the long pieces of DNA, they have to drag a whole long strand behind them to get through the obstacle course. So it takes them longer. So towards the positive end of the machine, we have smaller pieces of DNA. So you can see I'm pointing them to the bottom right of the gel here. So these are smaller pieces of DNA. And the pieces that are closer to the top are longer. They're bigger pieces. They weren't able to make it through the obstacle course to the positive end. Okay, so you might be wondering, okay, why would this be useful? Why would getting DNA, cutting it up, and running it through a machine so there's different sizes, like why would I even want to do that? So there's a couple different uses for this test. The first one is paternity testing. And so essentially if we have, let's say that we've got a horse and we know the DNA of the mother of the horse. We've got the baby horse, the foal, but we are not sure which father is the father of the horse. We can take the DNA from the possible fathers, we can take the DNA from the mother and from the baby horse. We can run them through a gel and we can see, are there any matches? Obviously, the mother is going to have matches or bars that line up, and then we could look through the possible fathers and see, well, hey, of the bars that are left, are there any matches? And if there are lots of matches, then, they'll say that this is probably the father of the horse. Okay, similar scenario with a crime scene investigation. So if we find DNA at a crime scene and we don't know, which of the suspects is guilty, we can run the crime scene DNA and then run the DNA of like all of the suspects, and whichever bars match up is going to be the person who was there at the crime scene. We can also use this technique to study the diversity of populations. Remember, we've learned before that more diverse populations are actually better because they're able to adapt to change because there's more traits to choose from or more traits that can respond. And so this technique would allow us, if let's say we tested a whole herd of deer, we could see, okay, well, where are the different bars? Is there a lot of different bars, or are they all lining up? Are all of these related? Is there not a lot of diversity? Okay, so here you could see we've got the results of like a paternity test. So here's the female, the mom that we were talking about. We were talking about horses earlier, but maybe this is people. So here is the mom. Here is the infant, and then these are the two possible fathers. So how do I read this diagram? We need to find the places first, the DNA that we know. So the DNA that probably came from the mom, because we know that this is the mother of the infant. There are some bars that match. This is DNA that probably came from the mom. So these are specific genes, maybe for hair color or eye color, that came from the mom. And so we want to rule those out first. And so this first bar of right here, this first bar is the first kind of match that we have with the infant. And so this piece of DNA probably came from the mom. The next bar that we've got that's matching up from the mom is right underneath the first, right? So this second piece of DNA for the infant also probably came from the mom. Okay, then we've got some gaps, but we're still focused on mom. And we've got another bar or another match right here. So this piece of DNA or this gene probably came from mom. And then finally, we've got one more piece of DNA that likely came from mom because there's a match. So that means that below the two, we've got two pieces of DNA that we can work with for, maybe they came from the potential from the father. This piece of DNA must have come from the father, and then this piece of DNA came from the father as well. So I'll go ahead and use a different color for the piece of DNA that probably came from dad. So we'll use blue. So let's take a look at this first one. So male number 1 does not have this piece of DNA. And remember, the DNA has to come from somewhere. So it had to come from mom or it had to come from dad. But male number 2 does have that piece of DNA. And so this is a piece of evidence that male number 2 might be the father. Remember, there could also be another father at play that we don't know about. Okay, so let's keep going for that reason to get more evidence. All right, so this next bar of DNA, you can see that both of the fathers actually have it. And so this piece of DNA, could have come from male number 1 or male number 2. And so this piece of DNA really doesn't give us a lot of information about who the dad is. The first piece was much more helpful. All right, let's keep going. So now let's head on to bar number 3 in the infant, and you'll see that again we have a match for male number 2. So this piece of DNA could have come from male number 2. And then the last bar, bar number 5, you can see this is inconclusive. It doesn't tell us whether it came from male number 1 or male number 2. So let's tally up our results and see, who is probably the father. So you'll see that male number 1 has 2 matches, 2 DNA matches that can be accounted for, 2 bars. Okay, whereas male number 2, if we look at male number 2 over here, there is 1, 2, 3, 4 pieces of DNA that match. And so we would probably err on the side of, how they usually say, we're 99% sure, right? There's a certain percentage, but we're, sure that male number 2 is probably the father. Male number 1, there's not as much match. And then also there's some bars that are not accounted for. Remember this first bar that we looked at? It has to come from somewhere. And so it did not come from mom. And it didn't come from male number 1. It had to come from mom or dad. And so that kind of eliminates male number 1. Another thing that you might have noticed was, hey, what about this second bar? Male number 1 and male number 2 had it. And so maybe male number 1 and male number 2, that particular gene is similar between them. And so that's why it would give you that matchup. Maybe they both, male number 1 and male number 2, maybe they both have brown eyes. So that's how you would get that kind of matchup. Another thing that you might have noticed as well is that at the top bar, male number 2 also has this gene that the female, the mom, has. So let's say that this is brown hair, okay? this gene maybe, and the infant clearly has the trait, like let's say it's brown hair or that piece of DNA. So it could have come from this female and it could have come from this male. We don't know. We're really looking for genes that are, or pieces of DNA that are unique, that give us information. All right, we're going to do it all again. Looking here at some crime scene data. All right, and so you'll notice that this is some DNA from the crime scene, and then we have those 3 suspects. We're not sure, if who was there, who was present, and you could see that if I look across, I know it's highlighted, the answer is given to you, but if you look across, there's a 100% match of suspect 2 and the DNA at the crime scene. All of the DNA that was at the crime scene came from suspect 2. There's some matches, right, in suspect 1 and suspect 3. Some of those do match up, but there's a 100% match between the crime scene and the suspect. So we want to account for as many bars as possible, because remember, the DNA has to come from somewhere. All right, so we are in your notes. Sorry about that. All right. Okay, so now it is time for a quick check. So in your notes, we're on page 7. This is where I am expecting you to write your answer to this quick check. So here we've got a mom and a dad, and then we've got 4 children, okay? Child 1, 2, 3, 4. And we are asking you to look at the DNA and determine for each child, is this the biological mother and father of all of these children? For some of them, is this the mother? For some of them, the mother only? Is this the father only? Or is this not the biological mom or dad of some of the children? And so take a moment to, again, on page 7 of your notes in that column, right? Write your answer to this quick check. Right. So if you take a look, this example was really nice because it's color coded, right? And so let's go over the quick check answer. So, check what you've written in your notes here. So child number 1 and child number 3 are the biological children of this mother and this father. Child number, and that's because all of the DNA is accounted for, right? Like all of the bars either came from this mom or this dad. Okay, child number 2 is the child of this mother, but not the child of this father. She has other bars of DNA here, they're in red, right, that do not come from this dad. The first bar, right, this red one, it does kind of match with the dad. So you might say, oh, well, maybe this is the father. However, the second red bar is it's a no match. And that DNA has to come from somewhere. So this is not the father. And then child number 4 doesn't have any bars that match. And so maybe this child is adopted. All kinds of families look different. It doesn't mean that this mom and dad, don't have an awesome family and they don't all love each other. They're a blended family, which is awesome. Okay. Our last DNA technology tool and technique that we're going to be looking at is called CRISPR. Kind of sounds like crispy. So CRISPR is a tool that allows scientists to really precisely cut DNA at very specific locations and edit it. And so it uses kind of a team of RNA, we've learned about RNA, right, and a protein called Cas9 to cut the DNA. It's even more specific than restriction enzymes. And it's a really cool technique that can be used to actually, cut out a diseased gene. We learned a lot about mutations and genes that can be, a mutation happens and that, person then might have a disease. Well, this technique can allow, the clinicians to cut out the piece of DNA that is diseased or that caused the person's disease and replace it with a healthy gene. It can also target a lot of genes at once instead of going one by one. And this is really, the future of health. So I'm going to show you a quick video on this technique so you can see it in action.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
So that was a really awesome overview of how this works. You might be wondering, well, why is it called CRISPR? That sounds very crispy. It is an acronym, of course, and essentially, as you saw in the video, it works like a scissors, and it's able to cut that DNA, and then we can insert new genes, or we can learn about what does that piece of DNA do, because it won't actually work if it's cut, because it tries to fix itself. So again, you saw this in the video. We've got the Cas9 enzyme, remember, which is a type of protein. We've got that piece of RNA that matches the targeted DNA, the section. So CRISPR then cuts that DNA at that very specific location, and then scientists can edit that DNA, remove portions that are harmful, or add genes that are desired, whether it be like you saw in the video, to engineer specific animals for specific traits, or to cure disease. All right, so that is it for our notes packet. We are finished with our notes for this unit, and so next class you should expect the test. All right, so how do you prepare for that? Well, we do have an assignment for you to complete today to practice more gel electrophoresis interpretation, as well as all of the study materials that are on Canvas. So please get all your work done today, and I will see you next class.